All right. Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. I'm joined today by staff members from each center, including my colleague here at NCCAT, Ed Eng. Joining from the Stanford SLAC CryoEM Center, S2C2, are Nathan Burrows and Michael Schmidt. And from the Pacific Northwest CryoEM Center, PNCC, is Sean Mulligan. And before we give the floor to our speaker, we'll get a quick update from each center on the resources we make available at no cost to the research community. For today's main event, PNCC has invited Joyce Fung, who's joining us from University of Texas Southwestern Medical Center, and Sean will introduce her properly after our updates from the centers. This webinar is a regular event. We host the last Thursday of each month, highlighting users of the National CryoEM Centers who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research. You can save the date already for our talk on February 26th. NCCAT will be hosting Vladimir Kolyadko from the Children's Hospital of Philadelphia, who will be giving a talk on cryo-EM of liposome-associated macromolecular complexes to reveal protein membrane interactions in hemostasis. So that should be a really interesting talk with some fun processing tips for liposomes. Registration link is already on the events page at cryoemcenters.org, where you'll also find recordings of all of our previous talks. And if you want to see us in person instead of here on Zoom and are attending the Biophysical Society annual meeting in February in San Francisco, you'll find staff from the centers there. Certainly, CryoEM Subgroup Saturday is a great place to find us, or in the exhibit hall at booth 410. So come see us or send your colleagues to come meet us and find out how you can make use of our CryoEM resources. For final logistics today, if you have questions about accessing the center resources, please feel free to reach out to any of the panelists here during the talk using the chat. And for questions for our speaker during the talk, please use the Q&A box. Sean will moderate the Q&A at the end of the talk using the questions submitted through that Q&A box. So let's hear what's new at all of the centers. And to start, I'll turn over to Ed. Let us know what's happening at NCCAT.
Hello, my name is Ed. NCCAT offers high-end data collection services, training, and grid preparation and screening services. We offer nationwide access at no cost at the point of access. If you want to learn more, you can go to our website. Putting on my outreach hat, I am the BPS CRYOEM subgroup secretary and treasurer. So if you are going to be at BPS, don't forget we do have a subgroup dinner. Tickets are available through the website at BPS. Also, I'm pleased to announce last year, the Agron Institute gave the CRYOEM subgroup a one-time grant to fund specific research meeting and symposia focused on cryo-EM studies. So if you want to learn more, you can always inquire with me about the subgroup or about the Agron Institute's grant. Thank you.
Thank you, Ed. I'll turn over to Mike or Nathan. I'm not sure which of you is giving the update.
Well, I'll go this time. I'm Mike Schmidt. I'm one of the co-directors of S2C2. We have 4 high-end Krios microscopes plus Glacios and a Tundra and all the specimen preparation equipment that's involved for service and for training and screening. A an additional perk we have for trainers and trainees and screeners is that Nathan runs a peer group discussion where people can come with their image processing problems for us, and that's held every week. And except for our Krios microscopes and our other microscopes, it's relatively warm here. So come see us.
Thank you, Mike. And now I'll turn over to Sean. Let us know what's new at PNCC.
Thank you. Hi, my name is Sean, and I'm a microscopist at the Pacific Northwest CryoEM Center in Portland, Oregon. PNCC accepts 2 types of proposals, screening access and general access. Screening access proposals allow you to come on site for up to 5 days to work on sample preparation and screening with PNCC staff. This proposal is aimed at low resource institutions. General access proposals award up to 480 hours of microscope time a year for 2 years, a total of 960 hours. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover a variety of topics in the CryoEM workflow. Approved proposals are also assigned a scientific point of contact, or SPOC, who can assist with questions about screening results, sample optimization, data collection and processing strategies, and so on. Our center has an Arctica, a Glacios 2, and 4 Cryoses. 2 of our 4 Cryoses are configured with K3 detectors and biocontinuum HD energy filters. The other 2 of our Cryoses are configured with Falcon 4i detectors and a Selector X energy filter. For sample preparation, we have a Vitrobot, a Leica EMGP2, a Vitrojet, and manual plungers. If you're interested in applying, our deadline is the 1st of every month. Please visit our website for more information, and we look forward to hearing from you. And it is my pleasure to introduce Dr. Joyce Fung, an assistant professor at UT Southwestern. Over the past decade, Joyce has focused on nuclear export, beginning her work as a graduate student using X-ray crystallography in the lab of Dr. Yu-Min Chuk, followed by a 2-year postdoctoral fellowship at the University of Pennsylvania in the lab of Dr. Elizabeth Rhodes, where she trained in single molecule fluorescence. In 2019, Joyce returned to UT Southwestern and the Chuk lab, where she started using CryoEM. In 2024, she joined the structural biology laboratory as an assistant professor, where she now uses CryoEM, microED, and small molecule crystallography while supporting a broad range of research and a lot of teaching. With that, over to you, Dr. Fung.
Thank you, Sean, for the nice introduction. And thank you, everyone, for tuning in for my talk today, titled Studying Exportin 1 by CryoEM, Revealing the Allosteric Mechanism of SINE-induced Degradation. Okay, so I would like to start with a acknowledgement slide. So this work is published last November, and this was in Nature Chemical Biology. And this was really a culmination of 12 years of work in Yu-Min's lab. And I started this when I was a graduate student together with Toga, a previous member of the lab, where he did the cell work and I did the structural work on this project. And he kept this project going for 10 years until he left, and it got passed on to Casey, who finished up this work. This is a collaboration with another 2 more labs, where at KU11, our collaborators, the Dalman's lab, Bert is one of the co-first authors of this paper, where they did a lot of the cell work to finish up this project nicely. And Yu-Min's longtime collaborator, Rosa, at Ohio State, who had data on patient cell lines. And in addition to all the authors in this paper, I also need to thank the cores at UT Southwestern that really helped make this happen. The SBL, the staff James, Yang, and Yan, our local CryoEM facility, where a lot of data is taken. And also MBR is where we do our our solutions biophysics. And the high throughput screening core facility, which will be mentioned again later. And Yu-Min has a PNC's proposal, and Sean is our SPOC. So I really want to thank him for his help, and also for this invitation, enabling me to talk about this work myself on this platform. And also Kirsten for everything that she does. And as well as all the funding sources that made this happen. Okay, so let's get started with the introduction on nuclear cytoplasmic transport. So it's mediated by a family of proteins called caroferrin betas, or CAPS for short. And these proteins use a RAN-GTP gradient to ensure directional transport. So in the nucleus here, in the cytoplasm, let's start with this. There's RAN-GAP that is on the nuclear pore complex on the cytoplasmic side. And there's also RAN binding proteins such as RAN-BP1, and both of these help the hydrolysis of RAN-GTP to RAN-GDP. So in the cytoplasm, it's mostly GDP state. Whereas in the nucleus, we have the action of the exchange factor RAN-GAF, which is tethered to the chromatin. This ensures that RAN-GDP is exchanged to GTP, so it's GTP rich in the nucleus. And these caps use this gradient to do directional transport. So there's importance that goes in, there are biportins that goes in and out. But today we're going to focus on exportins. And so exportins are the ones that form a trimeric complex with strand GTP and export cargos in a cooperative manner. And now this is a functional trimeric complex that can translocate across the nuclear pore to the cytoplasm, where the action of RAN gap and RAN BP1 causes the hydrolysis of the RAN to GDP. And now the trimeric complex will fall apart. The cargo stays in the cytoplasm. The exportin comes back in for another round of export. And exportin 1 is the general nuclear exporter in the cell. It exports hundreds of broad functioning protein cargos and many different RNAs through protein adapters. Expo1 recognized its cargos via a leucine-rich nuclear export signal that was first discovered in 1994, 1995. And the first structures of Expo1 was solved by crystallography in 2009. Dong et al. Is a Yumin's paper, and Monarchy et al. Is from the Gerlich and Figner labs. I'm showing you here the structure from the Monarchy et al. Paper, where this is a mouse Expo1 bound to RAN GTP and Exportin, which is a cargo. So Expo1 in here in gray is a heat repeat protein, has 21 heat repeats. These are pairs of helices, A and B helices that stack to form a solenoid structure. And Expo1, when it's bound to cargo, it forms like a ring. And so the RAN GTP binds inside in the inner surface, and Exportin uses the outer surface, the outside. If we zoom into the NES, we'll see that the NES has 5 hydrophobic residues that inserts into 5 hydrophobic pockets in the groove formed by heat 11 and heat 12 here in gray. And so over the years, and this includes my graduate work, the consensus of the NES has really expanded to a total of 11 different patterns of the hydrophobic residues. And the hydrophobic residues is no longer just leucines. It could be leucines I, V, F, or M. And solved by crystallography, many different NESs bound to Expo1. And the NES structure itself can be very diverse, but the groove has always stayed the same. And I want to reiterate the concept of allosteric for Expo1. So this groove, the opening and closing of this groove is entirely allosterically controlled. And what do I mean by that? So normally when it is on its own, it is somewhat auto-inhibited. So the groove stays closed. But there is affinity of the NES itself and the RAN GTP itself as well. And when both things are present, they bind favorably. That's the positive cooperativity that I mentioned earlier. And how does the binding of the individual component achieve this? And it's because there are 2 allosteric switches that really enable them to couple conformational changes to the groove opening and closing. So if we look at this unlinked CRM1, this is a thermophilia homolog for Expo1. You can see in the structure that the heat 21 has a long B helix that actually transfers across the solenoid and positions a loop that interacts with the bottom of the NES groove. There's also a long H9 loop that's a hairpin that also packs in the back of the NES groove, keeping it closed. But when RAN GTP has to bind in the middle, sterically, it pushes the 21B helix away. So it reconfigures to now interact with the rest of the heat repeats normally, effectively bringing the ends of the terminal and C terminals closer together to close the ring structure of Expo1. And the H9 loop also moves away from the back of the NES groove and kind of in a seatbelt configuration binds to RAN GTP. And so both conformational changes now allow the NES groove to open and bind an NES. So that's how the binding of the 2 is allosterically coupled. And we'll get back to this later. Okay, so now let me take a step back again to talk about the cargos. So Expo1, as I said, export tons of cargos, many of which are tumor suppressor proteins or pro-apoptotic proteins. So one might actually think that this would be could be used for cancer therapeutic because the mislocalization of these proteins actually confer survival advantage for cancer cells. And in fact, overexpression of Expo1 in cancer cells is often linked to poor prognosis for various cancers as well. The field has always tried to inhibit Expo1, but the first compound called leptomycin B, it's a natural product from Streptomyces. It inhibits Expo1, but it turns out to be way too toxic and failed the first preclinical trial that people tried to use this on human cells. So later in 2012, Karyopharm designed some small molecule inhibitors called SINEs, which turned out to be better tolerated. One of which, called selinexor, or the trade name Expovio, is approved for diffuse large B-cell lymphoma alone, and as well as in combination with bortezomib and dexamethasone for multiple myeloma, and the approval came in 2019. So how do these inhibitors work? They work by directly inhibiting the Expo1 NES interaction by conjugating to the NES group, actually covalently. So these are crystal structures that were solved back in, I think, 2012 in Newman's lab. And leptomycin B is a long polyketone, and it uses the double bond here to form a microaddition with a cysteine that is on heat 11A helix. It occupies the majority of the groove when you compare it to the important on the left here. And KPT-185 is one of the SINE compounds, also works the same way. It has a double bond that conjugates to the cysteine. As you can see, it's smaller, so it occupies only the top part, leaving pocket 1 and pocket 0 of the NES group open. So the KPT-185 is used kind of interchangeably with selinexor in the lab. They're very similar. You can see the top part is a little bit different and the bottom part is a little bit different, but we use it interchangeably in the lab. So what is special about these SINEs is that in addition to Expo1 inhibition, people have observed that it caused Expo1 degradation. And that is not the case for leptomycin B. So Tolga in the lab has found that this degradation is proteasome mediated because see, here's the baseline. When you treat cells with selinexor, the Expo1 levels decrease. When you treat them with selinexor and the proteasome inhibitor, MG132, you can see the levels are restored and there's a formation of a higher molecular weight band. If you inhibit this with a nedylation inhibitor, MLN4924, you see also the Expo1 levels are restored and there's no modified Expo1. So this process is proteasome mediated and nedylation dependent, meaning that it's very likely a Cullin-RING ligase system that is mediating the degradation. So we as structural biologists looked at this and be like, what is different about this that is causing this degradation? And the most striking thing is the empty P0, P1 pocket. So we thought, oh, is this the 3D diagram that's being recognized by the system to send it to degradation? So we tested this, and I did this back in 2014 with crystallography, because that was the method that we could do at the time. And I tested this two ways. One is by using a longer inhibitor. So we found this KPT-396, which has a longer extension on the oxygen compared to the 185 here. And I saw the crystal structure and it binds the same way. I didn't see any density for this extension, but it's likely just flopping around. But just having this extension is sufficient to block degradation in the cell, as Tolga has shown here. So what about messing with the group itself? So I also did that. I mutated this pocket to remove the hydrophobicity. So I included a salt bridge with the histidine and the E here. And both side chains are well resolved in the map as seen in the crystal structure. And indeed, when Tolga expressed this mutant as a flag tag protein in the cell, he could see that the flag tag signal is not reduced upon treatment of KPT185, whereas the endogenous Expo1, which doesn't have the mutation, would still be degraded, showing that disruption of this pocket can also block degradation. So it's likely the 3D diagram. So we know that from 2014, but we did not know what is recognizing that. So Toga went on a 10-year hunt using a genome-wide siRNA high-throughput screening, and this was done in collaboration with the screening core. And his assay was looking for degradation. So the readout was the Expo1 levels. And while we got the list of putative targets and trying to validate them all, the Dahlman's lab found the ASV8 as the ubiquitin ligase that actually promotes the degradation. And they used an entirely different method. They had a CRISPR screen, and they weren't even screening for Expo1 levels in the beginning. They were screening for selinexor sensitivity, so how effectively selinexor is killing the cells. And when we saw this and we looked back at our target, we're able to piece together now the machinery that's likely the thing that's responsible based on the data that we had. So it's likely a Cullin-5 system, which uses ELO-BC to recognize the substrate adapter ASV8, which would recognize the SINE conjugated Expo1. And Cullin-5 uses our RBX2 to recognize the respective E2s that would be responsible for this process. So we actually reached out and we formed a collaboration, and we were able to wrap up the story and publish together. Okay, so the first thing that we needed to do then is to make sure that this is indeed the architecture that binds SINE-Expo1. So Casey obtained a lot of these constructs from the Comab's lab. We have to thank them. They work on Cullin-5 and ASV9. And so Casey tested this first on a pull-down assay to make sure that the ASV8 is able to recognize the Expo1. So we routinely do pull-down assays. This is on a GSH beads. So this is the GST background for interacting with Expo1 with or without treatment of the inhibitors. This is a positive control where the GST NES would pull down Expo1 effectively, and it's inhibited effectively by both ABT185 and leptomycin B. And now the last 3 lanes is GST ASV8 along with ELO-BC. Casey found out that she has to purify them together. And this protein is able to pull down Expo1 only when 185 is conjugated, but not leptomycin B conjugated Expo1. So it looks great in vitro. Bert helped verify some of these components in cells to make sure that this is right. And so he used CRISPR knockouts. And you can see here, if you knock out ASV8, Expo1 level is not decreased upon selinexor treatment. UBE2R2 is one of the possible E2s, and the levels are slightly restored. ELO-BC, both is necessary for Expo1 degradation in the cell. And REH2 is a known E3 adapter that actually primes ubiquitin for Cullin-5 systems. So this also supports our concept that it's our hypotheses that it's Cullin-5. Okay. We also checked that in our test tube, we were able to actually target Expo1 for ubiquitination. And I have to thank the Commons Lab for a lot of these, and also thank Ning Zhang for a lot of constructive advice on how to do this properly. And so what I did here is I omitted individual components of the reaction, such as ubiquitin, the E1, the E2 for the RH2, the metallated Col5, the ASV8 adapter, and ATP, the substrate. And as you see, when everything is there, I can see a modified Expo1 band, but when any one of them is gone, that does not happen. And this shift is smaller than what a GST fusion would be, so it's likely just a single copy of ubiquitin. And I also tested in the test tube that we could actually get this to be polyubiquitinated. And so when we include E2s that are commonly used for polyubiquitination, with or without the RH2 priming ubiquitin, we are able to see some polyubiquitinated Expo1 in the test tube. So all this is to say that we have a nicely purified complex that is functional in the test tube. So how did we do this structurally? And obviously it's with CryoEM, that's why I'm giving you this talk. But I do want to stress that this was most structural studies on Expo1 has been limited to X-ray crystallography really until now. And we had to use yeast or thermophilia homologs with mutations in the groove to make it look more like the human version. And in many cases, the crystallography constructs are heavily engineered to really trap the conformation to help the crystallization to happen. For example, the structures that I showed you earlier were done in this kind of intermediate complex where we have Expo1 bound to RAN-BP1 and RAN-GTP, and it has a loop mutated, and the groove residues are mutated to look more like human. But CryoEM allows us to use for unmodified human Expo1 without any of this problem. So it was really nice that we could do that. And thankfully, thanks to that, we were able to have this paper in 2025. And there's another group that also has an Expo1 paper, also came out at the same time. So when we first did this, we were actually initially worried that it may be a little bit small. So the first thing that we did was just to try Expo1 itself with or without selinexor to see if it would work. So Expo1 is only 120 kilodalton. I mean, now with all the technology improvement, that is not small at all. But at the time when I was doing this, we were worried. But what I did was I buffer exchanged it into buffer without glycerol. It's previously frozen in glycerol. And then I screened it at high concentration with 5 different detergents. And this was the strategy that was picked because it worked for another protein in the lab. And I just used regular quantifoil, R1.2, 1.3, copper 300 grids. And I actually got good looking grids the first time. And so for the Expo1 itself, it worked with tioxopol. And for the selinexor, where I just incubated the inhibitor in 2:1 ratio with the protein, it worked with tween. And we collected data for the Expo1 and the Falcon 4 eye camera, and this was a K3 camera data set. So the data processing is nothing out of standard procedures. This was the first data set that I did in CryoSpark for this project. There was 8,000 movies collected. I did block picking, and then I did 2D classification and ab initio. And the striking thing here is that about 50% of the particles looks like broken up particles. So here is a complete Expo1. You can see here it just looks like this half of the protein, and then this would be the other half of the protein flipped around. And you can see this in the 2D classes as well. Like these are small broken up pieces of the big one. And this class looks like partially aligning both the broken up pieces and the not broken up pieces. And so what I tried to do was to then use the good ones and do topaz and see if I can enrich the population of the not broken up particles. And it didn't, I did it 2 times. It didn't really improve much. It still stays at 50%. But regardless, I was able to get a map that was 2.93 angstrom that I was satisfied with. And the same thing happened with the selinexor data set, where again, here, if you look at the ab initio classes, about only 50% of them is intact. 1 round of topaz picking, I think, got me more particles, but still. And this was at a 3.4 angstrom map. So when I saw this, I was like, what is happening? Let me try in solution to see if it's prone to being broken up, which I didn't think was the case. And indeed, this is in the condition that is in the grid, so no glycerol. I left it on the desk for 48 hours, and I don't see anything that is more broken up in solution. And so this was really my first personal and I think really drastic and kind of very clear case that the CryoEM grip prep can enrich monopopulations and also possibly even cause unexpected proteolysis. So I do not know what is causing this. There's already detergents in this sample that So I don't know whether it's the air-water interface or is it actually the mechanical force from blotting. So it'll be really cool to try this in a case that we don't have to blot to see if that would still be broken up. Okay, so since it's a CryoEM talk, this is my obligatory slide for my local resolution colored map for my two maps. They're both well resolved for most of the map, besides the end terminal, which is we know is flexible in this case. The map and the model overlay is over here. So let me now focus on the unligated structure first. So I do want to show that both of the allosteric regions that control NES group opening are well resolved in this CryoEM map. Here's the model, and I'll color the 21B here in yellow. And you can see the helix and the loop is well resolved. The H9 loop hairpin over here is also well resolved, binding to the back of this NES group in blue. And indeed, the groove is closed in this map. Here is the map quality for you to view on the left here. We have colored one side gold and one side cyan, and the back helices are in gray. So if the groove were to be a little bit open, you will see it peering through a little bit. So this is the crystal structure that was solved for human, but it was low resolution at 4.5, and it looks very similar, as well as the two homologs. One is thermophilia and one is yeast. They are all the grooves are close. So it's really nice to see that the cryo-EM structure is exactly the same as the crystal structure. And that is also the case for the inhibitor conjugated structures, most of it. So here's a crystal structure for selinexor bound to a yeast Expo1 that was modified to look more like human. This was really high resolution at 1.94, and you can see the inhibitor is very well resolved. In our cryo map, the selinexor does not have density on the top part. Oh, sorry. This cryo map has no RAN BP1. So it is free of crystal contact. But the heat 11 and heat 12 is still positioned exactly the same way. And then the top of the selinexor here, the pyrazinoyl propene hydrazide arm, I do not see density for that in the map. So it's likely flexible, and that is different from the crystal structure. Interestingly, this pocket that we know is open in the crystal structure actually have density for I don't know what in here in the CryoEM map. I have left it unmodeled because I do not know what that is. But regardless, it tells us that there is a gap between these two helixes. So we have established that we can do now Expo1 in CryoEM, and we can get a good map and see side chains. So now we have to do the ASB8 complex. So Casey took this on, and she actually had to optimize the ASB8 construct to improve stability because it tends to aggregate, and it wasn't looking too good in the grids. So she screened for different truncations and found that if she truncate this N-terminal 16 residue that's disordered, and including only the end-chrome repeats and the soft box, that it would improve stability. And so initially, her strategy was to preform the complex in SEC without glycerol and use a condition similar to Expo1. And that would mean to concentrate the complex down to high concentration and use detergent. But she found that it looks like things are aggregating. So what she did was then she input enough protein so that she can get a complex looking really nice in the SEC, and the small fraction at the peak of the right here, starred, would reach 0.5, 0.8 mg per mil without the need of concentrating. And she would add a little bit of excess inhibitor before she aliquoted it and just froze it in the freezer until she's ready to make grids. And she tried different grid types. She did R21 copper 300. She also did R1.2, 1.3 copper 400, and even C flats to get a nice ice for data collection. And initially, due to during screening, we looked at these images and thought they look kind of small, and we were worried for a while that maybe the complex was falling apart. But we decided to collect data anyways. because who knows, it may work. And in fact, it did. And this is the data processing that Casey did for the 185 conjugated Expo1 with the ASB8 with the N16 truncated with ELO-BC. So she did blot picking, and then she used the templates that she got from a subset for template picking, and again, 2D classification, heterogeneous refinement. And as you see here, we still have very small looking particles. That's why the screening looked very small. And there is still broken up CRM1, Expo1. If you look at this one, the Expo1 ring is only partial, and this one, the Expo1 ring is complete. And it's actually very obvious in the 2D classifications. This is part of Expo1 bound to ASB8 and ELO-BC. So it's honestly a little crazy that even if the ASB8 is present, there is still proteolysis of our Expo1 during grid making. Her map was 3.4 angstrom, and again, the core of the map is well resolved, and the termini of Expo1 is flexible, and that's why it's poor in resolution. So she did, then that's on the top is what I just showed you, is the 185 conjugated version. She also did one with RAN GTP to try to stabilize the N-terminal of Expo1, and this time with selinexor, since that's the one that's in use. And they are both very similar, so I'm just going to focus on the top one first. So if we look at the interface of Expo1 binding to ASB8 and we open it up like a butterfly and color them respectively, so here is Expo1 in gray, and the residues there in contact with ASB8 is colored in cyan, and you can see it spans across heat 10 to heat 13. And ASB8, on the other hand, uses ankyrin 4 to ankyrin 7 to make those contacts. And here's focusing on the interface on the top, close to heat 10 and 11 first. And these are a lot of charged residues and also some van der Waals contact. And if Casey mutated these residues, she could abolish binding of ASB8. So here's the wild type, and here's the Expo1 mutant, and here's the ASB8 mutant, and there's no binding. What about the other side? The other side is mainly between a short loop in the bottom of ankyrin 6, VRGL, and it packs really close to the bottom of the NES groove and makes many main chain contacts. There's one side chain contact, which is between L199 with the hydrophobic groove here, and there's some charge-charge interaction on the distal end near heat 12. And there is really no contact between the inhibitor and ASB8. The closest it'll get is this 4.3 angstrom distance, where this arginine is well resolved and just kind of sticks up. This is technically the van der Waals contact distance would be 4, so it's a little bit longer than that. And the ASB8 is really angled away from the inhibitor. As you see, the distance up here is quite big, it's 12. This loop is critical because if she mutates it to VGGG to increase flexibility or start including bulky residues such as arginines, she would also abolish binding. The same EH mutant that I generated on the Expo1 side would do the same. If we overlay the ASB8 structure with my EH X-ray structure, you can see that there is no room for that leucine 199 to really pack into the pocket 0. These mutants were also tested in cells, and we could see that these mutants do not cause Expo1 degradation like the wild type as efficiently. Bert also tested these mutants in the selinexor sensitivity, and indeed the mutants do not kill cells as effectively as ASB8 wild type. There are more cell work that I could show you that I don't have time for today. You could look into the paper if you're interested. Okay, so it seems like mutations on both sides of the interface is necessary for binding. So what is unique about this pocket though? If we look at all the structures that is known So these are all structures of Expo1 bound to different things, cargo, even a NUP214. And you can see this side of the interaction, which is this side here. All the side chains that are used to make contact with ASV are in the exact same spot and is accessible. So the only thing that is not accessible all the time is this P0, P1 pocket that we thought is the 3D diagram. Because this pocket is usually closed when it's unligated, or if there's NES, it's covered, or when there's leptomycin B, it's also covered. So what this is saying is that these signs are really conjugating to Expo1 and revealing a cryptic 3D diagram, a binding spot for ASV to bind and cause downstream degradation. It doesn't make much contact with the inhibitor, and it's mostly there just allosterically to open up the group. So this is an allosteric mechanism. So let me elaborate a little bit more on that as well. So normally, and this is kind of a redrawing of what I showed you earlier in the introduction. So when there is no inhibitor around, the group will have an equilibrium to go from closed to open, right? So there's a minor population that the ASV may engage. And that's the same for this case. When REN is present, there is a minor population that may be able to engage ASV8. So normally, without the inhibitor, that there will be low affinity. And because it's low affinity, it's unproductive for downstream ubiquitination. But when the selinexor is around, it's able to conjugate covalently to the groove, keeping it open, driving the equilibrium towards the state where ASV8 can effectively engage in high affinity. This is in contrast with conventional molecular glues, because those often make many contacts with both the substrate and the downstream and the adapter for ubiquitination. So for example, the CR8 inhibitor is clamped by the substrate CDK12, and you see it remarks the contacts that it makes with the adapter DDB1. Same case here in IBG1 and MMH2 in the BRD4 DCAF16 system. So this is in contrast in our case, where in our case, like I mentioned earlier, the closest distance it gets is 4.3 angstrom for the 185. Unfortunately, the selinexor has a bigger motif in the bottom and actually gets in van der Waals contact, 3.5 angstrom, but it's only a single contact. So is this contact important? So the first thing that most people would do is to mutate that arginine to alanine, and that is what we did as well. We mutated it to alanine, and we saw that it does not affect pull down, and it does not affect the ITC affinity as well. Casey measured it; they're fairly comparable. And the reviewer asked us to solve that structure. And we complied. And ironically, this was actually the higher resolution map that we've obtained against this in CryoSpark. And this is when we learned that we should do more heterogeneous refinement. And Casey went throughout and did 13 rounds of that to clean up the particle stack. But she's able to get 2.5 angstrom out of this. And surprisingly, or perhaps we should not have been so surprised, that selinexor actually changed configuration in this structure. Without the arginine, there is now nothing in that cavity. And then so the ASV8 angled a little bit differently, and the selinexor arm that was flexible before has now completely turned downwards. If you compare it with the wild type situation, this arginine is slotted right there. So that is an unfortunate surprise, I think. And this is an artifact because this is a mutant. So that doesn't happen in the wild type case. So this is not a good way to test whether that contact is necessary. So then we took two different ways to do this. One is to just make the inhibitor smaller. If we are suspecting that this 4.3 could still have an effect, what if we make it even smaller, right? And another way that we could use, because we know Expo1 so well, is that what if we just don't even use the inhibitor and just use the allosteric switches that we know to drive the configuration, drive the conformation of Expo1 to have more open grooves to see if we can promote ASV binding. And that is what we did. So let me first go through the small inhibitors. We found this KPT-127 that, oh, I'm sorry, it's missing something here. But KPT-127 only has a chloride on one end. And then we also talked to our chemist at Southwestern, Joe Reedy, and he suggested we can make a 6-member ring without the chloride, a 5-member ring, and a 3-member ring. And so we tested these inhibitors, and Casey did the binding and saw that ASV8 binding could still happen with 127 equivalent to 185, as well as UT Southwestern 1, and it starts getting weaker with the smaller things. And it's very likely because these small inhibitors don't actually get on Exportin as efficiently due to a weaker K-on. There was also cells cell experiments to show that we see the same trend in cells. So we solved that structure of 127. Casey obtained this map. It is a slightly lower resolution, but she's able to see that the inhibitor binds as expected. And now the closest distance we get is 4.7. Okay. We actually did use Southwestern 1 as well, but that map is even worse. So it says the same thing. Okay, so we were able to achieve this by using smaller inhibitors. So how are we going to do the experiments here on the right without the inhibitor? So there's 2 ways. One is including a mutation in the H9 loop to dislodge the loop from the back of the NES groove so that the groove have more propensity to open. And that is a trick that I had to use for my crystallography. So I know exactly which residue to mutate to do this. Another way is to use RAN. We can purify RAN and RAN GTP form that's very stable, and we can give it a lot of RAN GTP to drive the conformation to a mostly open state. So that's what we did in the pull-down assays. And first, I want to show you that we did observe low affinity binding without inhibitor if we titrate up the Expo1 concentration. So here is the same pull-down assay with GST. This is the background, establishing background binding to Expo1 as we go up in concentration from 1.25 micromolar to 10. And we can do the same with GST ASV8, and we can see that there is visible binding at 10 micromolar. And Casey actually took the time to try to measure this in ITC, and she was able to get an affinity of 11 micromolar in the cold. So now we established that there is low affinity binding. What if we have RAN? And this is the case where we added now 3 times excess RAN GTP, and you can see there is visibly more binding of Expo1. And the mutation of the H9 is even more drastic when we have this V43D is the H9 loop mutation. And you can see in all cases, the Expo1 can bind to ASV a lot more efficiently, even without the action of the inhibitor. So we were able to show that the inhibitor, the single contact that we saw is not necessary for ASV8 binding. We were also even able to utilize the allosteric switches in Expo1 to promote ASV8 binding without inhibitor, in a way showing that this degradation is through an allosteric mechanism, unlike the known molecular glues out there. So that's it. And really I showed you a small subset of the data. There are a lot more data in the paper. And as I said, this is a 12-year work. There's a lot of data that did not end up in the paper as well. And I was somewhat emotional when this paper came out because this is something that I started when I was a student. So I really hope that you guys were able to, learn something from this, and I found it interesting. And that's it. Thank you. Okay. Let me see if we have any questions.
While we give people a chance to type questions in, because that'll take a second, I'll pop in with the first one. Um,
Mhm.
so I'm really curious in your, so you see these partial bits of Expo 1 and you think
Yes.
it might be degradation. Um, I guess one question would be like, can you tell between, or proteolysis, could you tell between proteolysis and unfolding? And did you ever try to reconstruct those smaller fragments? Are they low resolution? Um,
Yes. Yeah. Yes. So I actually did try to reconstruct them, and they look exactly like this. It's this spot, and this one is worse, but it's a bunch of small helices. I don't think it's not degradation because it's clearly just breaking into two pieces. It's quite crazy. There is a loop right around there, a long loop, and there's that loop. Actually, we shorten it for crystallography purposes. So I personally think that loop just got cut somehow. I don't know how. Um, but yeah, I think without the C-terminal helix branching here, maybe we would think that it's unfolding, but it's so well resolved that you can even see this piece completely lined up with this that I don't think it's unfolding. Yeah.
and did you try using the shorter loop construct in cryo-EM?
Oh no, we did not do that. Yeah, because we that construct was not we don't have that in the human version. I have it in yeast. So yeah, again, one may be able to try to throw yeast CRM1 in here and see if it would break it open. Um, it's kind of crazy because the ASV8 binds right there. So I thought there may be less lysis or less breaking of the particles if the ASV8 is present. Um, it I think there's maybe a little less. That could be wishful thinking, but it obviously still happens. So that loop is available for to be broken up somehow. Um, yeah, right. Detergent. So in my sample, there is detergent because this was high concentration, but Casey's sample, that was the complex, did not have detergent, and it's still the Expo one still fell apart. So it's not like the complex fell apart, but the protein itself, the Expo one, broke apart in two pieces, even in the case that there's no detergent.
Actually, I'm curious about your first approach was to just try detergent, and you said like this had worked for
Mm,
something else, so that's why we did
yeah,
it.
yeah,
I guess from your experience, do you think that's a good place to start? Like if you were starting a brand new project, would you
yeah.
just start with detergent right away? Um, or what's your opinion on that as a generalizable method to think about
Yeah, I
screening
think
for initial conditions?
this was really a special case because this protein is concentrated to a high concentration all the time. So we're not restricted by using any detergent. And that's why for these things, we always just screen 5 different ones and one or two of them will work. Um, I think having a little detergent did help with probably would help with the ice situation. At least in my case, it did. For Casey, when she couldn't use detergent, she had to try different grids, and it worked. For one of them, I think C flat worked. For one of them, it was the 400 that worked. So I think if you can get your protein to high concentration, why not try it? But obviously, I think it's different for different cases. This was complete just because we have crystallography grade protein, and I think it's having a good. This also is, I think, is a case where it speaks to the importance of how important your sample is, because it's purified so well that it worked the first time. But I think if it were to be a less clean sample, I would have to work a little harder at that.
All right. Well, if we don't have any other questions, we can go ahead and wrap up today. Thank you so much, Joyce, for sharing that story. Thank you to everyone for joining us. We wish you all as much success in your cryo-EM endeavors as Joyce has gotten to see. And we will see you here next month. Thank you.
All right, thank you.